Well, hey again, it's Mr. Bleeker for part two of enzymes. In part one, we talked about enzymes as these amazing little protein catalysts, these quaternary proteins that make our metabolism function. We talked about enzymes that can build things up and enzymes that can tear things down. In this second installment, we'll focus much more specifically on the mechanics of the enzyme. Enzymes are highly specific. Well, now it's time to find out exactly why that is. Now, this is one of the animations that I do have on the course site, and it's quite positive for several reasons. I'm really happy with how simplistically this demonstrates this, and it's very clean. So here we have an enzyme that is a protein catalyst, and we know that they're very finicky. They work only so well at certain temperatures, and they work well at certain pH levels. Okay? Yes, they're sensitive workers, so we need to pay attention to that because otherwise their productivity will be quite poor. Now, this portion right here, that's called the active site. Okay, that's where the business happens. It's where the substrate will bind. So, if we were building up maltose, for example, we would get a couple of glucoses to fit in here, let's just say, and we would be trying to build up. Now, an important distinction you should make is, well, what kind of metabolism is building up? Think about a really strong girl named Anna. We're going to build her up because building up things is called anabolism. I always thought it was a neat analogy, right? Building up Anna. So building things up is your anabolic metabolism. You've heard that term before. You've heard anabolic steroids. It's like, ooh, anabolic. Yeah, well, those are the kind of steroids that will build muscle mass and tissue density. The other side of it is catabolic. And my analogy in the previous lesson was think of a cat tearing up your couch. Okay, that's tearing things down. So that's the tearing down metabolism. So anabolism, building up, catabolism, tearing down. We need to look a little further now about enzymes and how they can do their anabolism, catabolism. We know that enzymes act on specific substrates. That substrate more or less has to fit. Enzymes can be fooled with some substrates, but we'll get into that in a minute. This is called the enzyme substrate complex, or the ESC. So what we get is a complete fit between the substrate and the enzyme. Whether or not that substrate is going to be broken down or built up isn't really the point. The point is that they fit together. So this is a catalytic cycle that we'll look at. So lactose. Well, you know this enzyme. It's lactase. For those of you who are lactose intolerant, you don't produce enough of this enzyme. And remember, lactose is a glugalac. There it is, glucose and galactose. Lactase is an enzyme that we have a lot when we're infants because breast milk is very high in lactose. So this is what lactase is meant to do, to liberate these nutrient molecules for us. So this is a catalytic enzyme. Okay, and then lactose just keeps coming and you process and produce lots of energy. I'll just go over here for a second. Just get this guy to play. There we go. So the active site here that's represented in yellow is, as I say, it's highly specific. It's not just going to fit with anything. Here's a neat little demonstration. Which one do you think fits? Oh, maybe this one. There you go. And there's this enzyme is catabolic. It's breaking down our products. You'll find enzymes all over the place. They're in our cell membranes. They're in our cytoplasm. They're outside our nucleus. We're producing them like crazy. Yes, they're made by your DNA because they've got so many jobs to do. That's why we have such an amazing amount of DNA. Now, we do have to talk a little bit about enzymes that can. There's a few things that you need to know. They can be. Let's go back one. Your enzymes themselves can be turned on and they can be inhibited. So let's just pause that there. Now, there are inhibitors. You've heard of things that can poison our enzymes. You might have heard of cyanide. Boy, that's a runaway. It just goes. I have to press pause. Cyanide can poison our enzymes and our mitochondria, which leads to death. Without ATP, you're just not going to survive. And there is lead, right? So I'm going to hit pause there. There we go. So with no inhibitor, things would keep going, but with an inhibitor, you've got a major problem. Your enzymes can't do their job. Now, just pause that. There are some things that can interfere with our enzymes. We've seen that, such as lead, such as cyanide. And we do use drugs to combat germs because we try to poison their enzymes so that their metabolism grinds to a standstill. Smart, hey? This is a neat little demonstration here. If you hit play, if you add an inhibitor, there we go. I think I'll have to forward that. There we go. Maybe it's on the next page. That was interesting. If we add an inhibitor to bacterial development, they can't grow. So if we take penicillin, for example, I'll hit pause now. Penicillin, I talked about this in Biology 11, will impede the growth of the cell wall. And if you remember, this cell wall here is made up of peptidoglycan. Well, penicillin can interfere with the enzymes that build up their wall, and poor little guys die without a cell wall. So yes, with no inhibitor, enzymes will do what they're supposed to. But with an inhibitor, enzymes can't really form their products. So yes, we can do enzyme warfare. And here it is. For example, this would be it doing its job. And here, there's a problem. Now, I'm going to pause that. Down here, what they're introducing, for example, is lead. Now, lead can be an inhibitor in a couple of ways. You can inhibit an enzyme by blocking its active site or blocking the back door. Some enzymes need a little bit of extra to help them function. Now, this is, you could think of this as a backdoor poison. I'm going to show you, let's see here. I'll show you this here. Some enzymes, in order to even function, will need an activator. Now, it kind of looks like a little key, doesn't it? Well, that activator could be a nutrient. For example, vitamin C. For example, some of the minerals that we consume. Now, this site on an enzyme is called an allosteric site. I'll pause that for one moment. So our enzymes may not even be functional until we have a key inserted. This allosteric site needs to be filled, or this enzyme will not assume the correct shape. That's a good way to regulate enzymes. You might want enzymes present, but until they're needed, they shouldn't be turned on. Once the allosteric site is filled, let's say that's a mineral like magnesium or calcium or something like that, at that point, your enzyme is functional and can do its job. It's a good thing. I'll just reset this animation. There we go. But sometimes enzymes can be inhibited, and until the inhibitor is released, then the enzyme's not functional. So this is kind of the opposite. If you think about this, I'll hit reset animation. We had to add an activator to what's called the allosteric site, and then the enzyme would work. Okay, that's just as nice twice. So inactive enzyme, that's activated. Okay. Again, this may be minerals, calcium, magnesium, zinc. Right? It's funny how this looks like the Death Star. Oh, now it's functional. When we look at, in this case, we've got an inhibitor and the enzyme can't even function. Now, sometimes we introduce drugs just to inhibit enzymes. Penicillin will do this, for example, with bacterial cell walls. And that's the whole reason we put that in there, was to put something in that would poison that enzyme, that would inhibit it. But sometimes enzymes, they're not poisoned at all. They're just, this inhibitor is just removed before the chemical reaction occurs. So it can't do its job until the allosteric site is freed up, and then the enzyme works. Okay. So if we go back and we look at this, the problem with lead is lead fits, it's what we call an inhibitor. And let's see, where is it? Oops, close that. There we go, enzyme inhibitors. So normally this enzyme will function really well with the addition of a material to its site. So it needs some kind of a cofactor, right? It needs a cofactor in order to assume the correct shape. So there's two ways to mess with an enzyme. One, you can plug its active site competitively, which is nasty. And the other way is what lead does. In a non-competitive way, lead doesn't go after the active site. Lead goes after the allosteric site on the back right here. Ooh. So I just have to replay the animation for you. Give it a second. There are some heavy metals that will literally just do this. Mercury, for example, will do this. But lead's a little bit different. It doesn't try to plug this site. It'll just try to plug the one in the back door, and now, oh, look at that. Lead can poison an enzyme into taking the wrong shape, so it can't do its job. So these are two distinctions you do have to make. Competitive versus non-competitive inhibition of enzymes. And just going back to this, that's what we're seeing here. Okay, so what type of inhibition is being shown here? This is non-competitive. Ta-da! There you go. Hey, I passed. Now, some of these inhibitors, some of these reactions, you wonder, is it reversible? Can this be undone? And the answer is sort of yes and no. Some inhibitors will let go, and others you can't get to let go. Heavy metal poisoning can be like this, and it can render the enzyme sort of ineffective moving forward. Okay. So certain drugs, when we look at this, we're looking at what they'll do to the active site, and we'll look at whether or not they're inhibiting in a temporary way or whether they're inhibiting in a long-term fashion. If we poison, if we use penicillin to poison our little friends, the germs, that's not reversible. But if we take something like aspirin or a painkiller, really what we're doing is we're turning off the enzymes, sort of the metabolic pathway that helps us to perceive pain. So we can turn that off so that we don't feel as much pain, and that's what analgesics do. All right. So I need to pop back to the notes here for one quick second. A little bit of note taken to fill in. There we go. All right. So, when we left the notes, we were talking a little bit about specificity, so I'll back up just a little bit. Lipids get torn down by an enzyme called lipase. So if we were looking at that, you'll notice that each of these enzymes tends to end in an ASE. Well, there's a reason for that. The way we name enzymes is we name them based on the substrate, and then we throw an ASE on the end to indicate that is an enzyme that's acting on that substrate. So maltase would break down maltose into a couple of glucoses. Urea is broken down by urease. Even RNA can be broken down by RNases, or in that case, a ribonuclease. It just depends on what we're looking at. Are we trying to build something up, right? You studied DNA polymerase. Well, okay, so that's an anabolic enzyme. Okay, so when we last looked at this slide, we were examining what enzymes are doing, whether they're building up or tearing things down. Okay, so we'll kind of go through a couple of these. It sort of given away. It says degradation, but let's look, sort of examine it briefly here. If our substrate pops into our active site, let's say this enzyme is specific enough to the substrate, what we're getting is broken up products. Now, when we look at that for a second, what we have is a catabolic process. It was broken down. Okay, let's look at the next one. In this case, if we're bringing in sort of raw materials like this, let's say we're bringing in glucose and galactose. If we bring them together with an enzyme like this, then what we're building up, geez, thank you, Etsy. Sure, I'll take $5, is we're building up a product. So this is an anabolic process, not a catabolic one. There we go. This slide, there's not too much to add to this, really, just to say that in this case, what we're looking at is we're looking at the ES complex. And I think students forget that you really have to know the terminology. So the enzyme substrate complex is that right there. That's where they're binding. Whatever the enzyme ends up doing is whether we're looking at catabolism or anabolism. That's it. So some pretty important things to remember. One, and I think it's been missed, is that enzymes themselves are very specific to a job, and they only really catalyze one thing. You can't expect them to do all sorts of things. They have preferred pHs and preferred temperature ranges. So let's make sure we know this. One type of reaction, very specific with the pH and the temperature. Right? You could denature enzymes very easily. You got to be careful with that. Enzymes, because they catalyze only one type of reaction, are really good at unitasking. They just do one thing, so that makes them very efficient. Like the robot in the factory that has one job to do. But nomenclature just means naming. So we don't have to read too much into it. It's just, how did we name them? So if you look at lactase, the reason it was named lactase is because it breaks down lactose. So I'll go back 3 letters there and throw in the ASE. So there you go. This is the enzyme that gives you glucose and galactose once it basically tears down lactose sugar. So some key points. Please remember, when we're talking about reactants, you call them substrate. Okay, if the enzyme is doing a number on something, call it this. The something must be the substrate, and the substrate is always the reactant. The active site is the point of business. Okay, that active site is right there, for example. Okay, and that produces your enzyme substrate complex. And there's a reason why that is a double-sided arrow, is because the enzyme substrate complex isn't meant to last forever. So although this is, if the enzyme is the lock and the substrate is the key, you remove the lock from the key, right? So it's a reversible reaction. So they call it lock and key theory. Now, it's not to say that enzymes, we did say they're super specific, right? And they only do one job, and they're only supposed to have one substrate. Okay, to be really clear about it, Enzymes have a preferred substrate. They're so specific when you look at their geometry, they don't fit with just anything. I mean, it's got to be pretty darn similar for the enzyme to bond with it. So, so that you understand, enzymes really just want a specific substrate, not just any substrate. It's an induced fit model. So, when you hear that enzymes only have one substrate, that's what they mean. They have one basic sort of fit. There are some things that can sneak into an active site that look like the substrate. Enzymes can be fooled. It's certain poisons can that work that way, right? And it's not good. That's so some heavy metal poisoning can interfere with the active site, but that's not what was intended. So, since you've studied about temperature and protein, everybody's cooked an egg. You know that high temperatures can denature an enzyme pretty darn quick. And enzymes can be denatured. They're very sensitive, so they want to work in an optimal pH. So don't cook them, don't chemically fry them. They don't like that. That doesn't work out so well for them. It's not what they want. The idea is just stop here. The idea with an enzyme. Let's see here. I don't want that anymore. There we go. With an idea with an enzyme is that without an enzyme, you just have like really you're going to expect lactose to just put itself together. So if these are like glucoses and these are like galactoses, they should just run into each other and form a chemical bond? I don't think so. I mean, just due to chance, maybe they'll bump into each other. Maybe a bond will be formed. I mean, watch that again. This is just chaos. This is why enzymes, as matchmakers, help chemical reactions speed along. Because with an enzyme, if they fit into the active site, the enzyme can expend just the tiniest amount of energy and induce the fit. So glucose and galactose come together to make lactose. This helps the reaction. It's not consumed by it. It's not a part of the reaction. It it just makes it happen. And in this case, the enzyme's acting in an anabolic way. Okay. Just going to hit play on this. Those enzymes, please note, sometimes the enzymes do need a little bit of help. So consider cofactors. Now, I said sometimes with enzymes, you need things from your diet, like minerals, like vitamins, to make enzymes just the right shape. Those cofactors can sneak into the allosteric site on the back and turn on an enzyme. Sometimes the cofactor, if it's like vitamin C, will actually become a part of the active site. There's names for these things. The enzyme, if it needs a little bit of help from a vitamin or a mineral, the enzyme's called an apoenzyme. But that's not so important. I just want to show you what happens when a little bit of nutrition from our diet, in this case, vitamin C, will literally become a part of the active site. So this is what vitamin C does. And vitamin C is called it's a cofactor, but it's more formally called a coenzyme. And once vitamin C gets into the active site, it makes the active site take the correct shape. Now, other cofactors like magnesium and zinc and things like that on the backside, they can activate the enzyme too. So, just sort of an interesting little distinction to make there. It's why it's important to get your vitamins. It's why it's important to get your minerals, because if you want your enzymes to work, well, they make the active site function properly. Good. I'll close those two. I think. There we go. Sneak back to our notes here so we can finish off just the sort of the writing material. Good. Okay. So don't mess with enzymes. Give them what they want for temperature and pH. It's important, just going back to how finicky enzymes are, to give them what they want. Let's change color. I'm a little tired of blue. Let's go back to purple. This enzyme, is peak activity is right here, and if you trace that down, kind of loosely there, this enzyme's kind of happy around sort of the 40 degrees Celsius sort of temperature. Okay, so that's its happy place. If you look at protein digesting enzymes, this one right here loves a pH around 2. Now let's think for a second. Protein digesting pH 2. Where would that probably be? Oh, in our stomach. So this is a protein digesting enzyme that does its best work at pH 2. And then there's this one right here that does its best work around pH of 8. Well, what I'll tell you is that pepsin is found in your stomach because it likes the acidic environment. Okay, so it likes that. Trypsin is found in your small intestine, where a lot of digestion occurs. That is, after your meal leaves your stomach and gets neutralized, right? Think about it this way. Pepsin will not be working in your small intestine. Your small intestine is a pH of 8. So you don't count on pepsin to do any more work once it leaves the stomach. Its job is done. Really neat graph of this one, right? Looking at different, this is why we need so many enzymes. Pepsin's busy beating up protein at a pH of 2. And trypsin, of course, is over here beating up protein at a pH of, 8, approaching 9. We get that. Just erase those two. And then salivary amylase, well, it's not even after protein. It's after starch. And salivary amylase really likes a neutral pH. Well, salivary amylase is found in your mouth. So you can just see how preferential all these different enzymes are. Let's see if I can shrink that down a little bit. So please be aware that enzymes need that little bit of chemical help. Cofactors tend to be inorganic, right? These guys down here. Whereas coenzymes like vitamin C, vitamin B, things like that, all our vitamins, are organic. You don't just find vitamins in nature. If you think about it, you get a lot of your vitamins from eating fruit, eating your vegetables. So they're organic in nature. But what they do is they make enzymes' active sites work properly. So eat your veggies. There is a point that the distinction here in point 9. How is it that enzymes know how to turn on and turn off? If you think about it, if a baby is consuming breast milk, how do all the enzymes know it's all hands on deck? Right? All of a sudden, there's this nutrient that needs to be taken care of. Well, that really comes down to enzyme regulation. And your body will produce more enzymes or activate more enzymes in the presence of their nutrients. So if there's a lot of lactose present, the nutrient itself can turn on the enzymes, is what I'm saying. And the enzymes are just sort of present until they're turned off. Once the nutrient is consumed, there's not enough to activate the enzyme. The enzyme itself will sort of plateau and finish off. I'll show you an animation of that right now. All these animations, I know, but it's quite a theoretical thing when you look at enzymes. So you want to see really what you're looking at. Okay, not that one. So in the presence, now this isn't taught in Biology 12 formally. It's actually in chapter 26. But this is the DNA that normally produces your lactase enzyme. And your lactase enzyme is normally inhibited. There's no reason to produce lactase if there's no lactase around. So there's a gene here that is preventing your lactase enzyme from being expressed. Okay, so all of a sudden, holy smokes, there's lots of lactose in our bodies. So this is really neat. Without getting into this, this thing is called the lactose operator or the lactoperon. Just the presence of lactose in our bodies as a nutrient, as it starts to show up, just the mere presence of lactose can turn on enzyme production, and I'll show you right here. Lactose, it's like they drag away the inhibitor, kicking and screaming. Now there's no reason for this gene not to be turned on. Well, this gene, ladies and gentlemen, makes a lot of lactose. So with no inhibition, guess what's going to happen? There comes RNA polymerase. We just did this. And this mRNA is going to help us make all sorts of enzyme. And once you have that enzyme, you'll be able to break down lactose. So feedback inhibition. Actually, this one, it's not my favorite. It's not the one I want to show you. It's pretty good, but I like the lactose operon a little bit better. Oops. All right. One moment. Good old AirPlay. There we go. Okay. So point 9 is kind of important. The concentration of the substrate itself can turn on the enzyme. And when the substrate concentration is depleted, the enzyme can be turned off. I think that's a really a good sort of start and end point. We'll be getting more into feedback loops, positive and negative feedback loops as the course progresses. But for now, we'll just say that it's a feedback mechanism. And this is a neat little sort of revisitation of what cofactors and coenzymes can do. Cofactors are inorganic. There we go. And this is supposed to say allosteric site, not allostatic. Now the allosteric site can be in the back door. It could be this little pocket right here. And sometimes the allosteric site can literally be in the active site right there. So in this case, you can see where instead of being in the back door, it's this cofactor fit right into the active site. Just think of the cofactor or the coenzyme as an activator. That's really what it's about. Activating the enzyme, getting it to take the proper shape. You could see why not getting proper nutrition, not getting proper vitamins can lead to disorders in this regard. And here they are. Scurvy is a vitamin C deficiency. Now, my dog's staring at me over here. She won't have a problem with this. You see, dogs and cats and things like that, they don't really need to consume vegetables. They have the biological ability to make vitamin C. So their gums aren't going to get bleedy and fall out, and their teeth aren't going to fall out. If you recall some of your social studies history, the early explorers were drinking boiled willow bark just to get enough vitamin C from the willow bark to last them through the winter. It also had some aspirin-like qualities to it, sort of a painkiller, right? Pellagra, if you don't get enough B3, you'll get pellagra, and you can see an example of it here. Rickets, I'll show you an example of rickets in a moment. Night blindness is an interesting one. If we don't get enough vitamin A, in the back of our eyes we have rods and we have cones. There'll be it's an associated problem where your brightness indicators, your rods, aren't functioning at proper capacity, so you can't see with any degree of efficiency in the dark. Vitamin K, it's one of the reasons why our blood clots. Vitamin K is pretty important. I should make a note here. E. coli in our large intestine makes gobs of vitamin K for us. It's why after long bouts of antibiotics that some people can experience some problems because without proper clotting factor, if you cut yourself, you'll bleed sort of in a way you wouldn't expect. Not like not bleeding like full blown hemophilia, but you don't produce scabs properly. So vitamin K is really important to the production of platelets. Now rickets, I wanted to show. There's not a picture there, so I wanted to demonstrate one. All right, let's give it a second. If you don't get enough calcium, enough vitamin D, rickets can be a major problem. And let's see if it loads. Come on, there we go. Just give it a moment. There we go. Rickets becomes a major problem because in the normal formation of bone, you need both calcium and vitamin D. And without it, you'll get the malformation of bony tissue. And it's a very big problem in the developing world without proper nutrition. And it can affect even the closure on the top of the head. So this is an issue that comes from something as simple as a vitamin D deficiency. Vitamin D, strong sources of that are milk. We make a lot of vitamin D in exposure to sunlight, but it kind of goes hand in hand with calcium. So, yes, milk, it does a body good. Okay, just about done, folks. When you look at enzymes, when do they sort of stop doing their job? Like they're little worker bees, but the reaction rate can only really go up to a certain point, right? If enzymes are expected to do all this work, the rate can only go up to a certain point and then while you kind of max out. Well, maybe you only have so much substrate to work on. So when an enzyme is absolutely saturated, every enzyme is doing a job, then you have maxed out the ability of the enzyme to work. You can expect those little robots, for example, at a Toyota factory, to work up to a certain level, but you can't just start handing them more material and say, hey, work faster. They're already working at max capacity, and max capacity is right here when all the enzymes and all the substrates are basically busy. The matchmaker is going as fast as it can. So that's called a saturated enzyme. And you've hit max efficiency. So you can throw more substrate at it. If this was lactase, you can't just add more lactose and just say go ahead and process it. That doesn't fly. That won't work. One more look at enzymes and how you can deactivate them. Competitive inhibitors. Um, Understand that active site can be fooled, right? Um, a bad case of this is where pets consume antifreeze, which is basically a metabolic poison. And if the antifreeze will taste sweet to them, so they'll be licking away at it. But what they're doing is they're blocking their active sites. Um, it's a nasty, horrible thing that's happened in the past. Cyanide in humans does this. Penicillin does this. This is a way of poisoning enzymes. This is called competitive inhibition. Non-competitive inhibitors, um, well, lead's a good example of this. Hooks on to the enzyme, maybe onto the allosteric site, maybe onto the back. That's one way of doing it. And if you change the shape of that enzyme, well, you can see that the active site here has been, had this configuration and now has been stretched like crazy. Well, what you've got is an enzyme that can't do its job anymore. So a lot of heavy metal poisoning, for example, lead poisoning would do this. The lead aqueducting in Roman culture uh, led to some pretty crazy behavior by the Roman rulers. And what was happening? Well, their enzymes were being poisoned, and their judgment was heavily impaired by the action of that. So it's why we don't want to add heavy metals to our environment. They go around and inhibit our enzymes. That's not good for our survival rate. So we have to clean up our act and not allow these non-competitive inhibitors, these inhibitors, the class, to poison us in this way. So the control of enzymes, the pace, we've mentioned more or less that as long as the substrate is kind of present, it can activate the enzyme. And the enzyme will go as fast as it can. Um, Activation, deactivation. There's a variety of ways to do this. I showed you the how our DNA can be turned on just by the presence of lactose. It can sort of uninhibit the production of lactase enzyme. So sometimes the nutrient itself can lead to the production of the enzyme. Well, that's good, right? The nutrient itself becomes a little bit of a regulator. But activating or deactivating the enzymes is pretty important to do. We don't need them to be going like gangbusters all the time. Okay. And I think, ladies and gentlemen, I'm just going to leave it at that. I think that covers our topic quite well. Um, that's been another one in this series, and thanks for listening. Have a good one.